Assalamu alaikum everyone. My name is Rumaan. I'm a second year medical student, and today's PAL session is going to be on control of breathing. Okay, so without further ado, let's get started. The table of contents is just the objectives that Dr. Shams gave. Okay, these are also the objectives that were mentioned in your curriculum, the one that your BLT sent in your announcements group. Okay, so, yeah. Okay, one of my seniors gave some really good advice. His name's Naeem. He said, um, no question. It's a stupid question. There are only stupid answers. So the only one that looks bad here is me. Okay. So yeah, ask any questions that you have throughout the lecture. Okay. So let's start. This is the first image that's in Dr. Shams's slides. I don't remember studying it really. Okay. It's too much info, but it's a good summary of everything that you're going to study today here. Okay. So let's start with this part right here. Okay. This is the cortex and the brainstem. These are basically higher center. Okay. So these control the rhythm generator. Okay. The rhythm generator basically generates the rhythm for your breathing. Okay. And as you can see, this efferent output, I hope you remember intro to nervous system from F&D. Okay, this goes down to your respiratory muscles. And then there are, receptors with or without feedback. So with feedbacks in blue, you have mechanoreceptors and chemoreceptors. We're going to talk about them. And without feedback, you have mechanoreceptors in musculoskeletal system. So those that you voluntarily control, okay? But these don't have feedback, right? So mechanoreceptors or chemoreceptors, they send afferent inputs, which means they are going towards the brain. You can think of them as sensory, basically, inputs, okay? They go to the rhythm generator. Something happens here. We'll talk about what that is. And then this goes to the respiratory muscles, okay? Whatever happens here will decide whether these are stimulated, inhibited, or whatever. That's why we call them with feedback, okay? These cortex and brainstem, we're going to talk about these, don't worry about it, okay? But you need to know afferent input, basically like sensory, okay? Efferent, more like motor. And yeah, we'll talk about these in detail, don't worry about it, okay? So let's start with efferent output, the one that we're talking about here, okay? These includes yeah, this efferent output includes the respiratory center nerves that go to the respiratory muscles. So basically accessory muscles that are used for breathing against resistance or exercise. So normally you won't be using them really, okay? And these are controlled by something called the VRG, the ventral respiratory group. We'll talk about those two. But accessory muscles are basically used for breathing against resistance or exercise. So when you want to breathe really, really quickly, okay, you don't give your times, you don't give your lungs time to like passively exhale. Okay, so you're going to be using expiratory muscles, inspiratory muscles. Those basically add-ons. You have expiratory accessory muscles, which include internal intercostal. The way I remember it is expiratory starts with an EX. It's going to be the opposite for the internal or whether external. All right. So inspiratory is external intercostal muscles. Expiratory is internal intercostal muscles. But you have abdominal muscles. In inspiratory, you obviously have more, okay? Because inspiration is normally an active process, while expiration is a passive process. Expiration basically depends on your lungs' passive recoil. I think you talked about this in one of your previous lectures. Okay, all good from this slide? I hope so. All right. So control of breathing, you have two main areas here. Okay, you have the respiratory center, mechanoreceptors, and the chemoreceptors. Let's start with the respiratory center, which basically includes the pons and medulla. This is parts in the brainstem. So these centers in the brainstem, these include a group of neurons that control respiration. Okay, so you have three groups of neurons. The ventral respiratory group, ventral for front, dorsal for the back, okay, and the pneumotaxic center, which is located in pons. You see, these two are in one color. This is in a different. I'm not colorblind, okay? These are here for a reason. I'm going to show you in a second, okay? Big diagram. We'll get back to this in a bit, okay? So dorsal respiratory group, this is located in the back, right? So posteriorly, I hope you know your anatomy, in the medulla, in the NTS. Remember where we talked about the NTS before in integration of cardiac output, the nucleus tractus solitarius? Okay, this includes the DRG and cranial nerves, okay? All right, so what's the function of DRG? integration of inputs, basically. So what this means is you get nerves that are coming into the DRG, okay? Whatever impulses they send, whether it's like really fast, really slow, that decides whether you're going to breathe faster, breathe slower, okay? This does this with the VRG. We'll talk about that in a second, okay? But basically, that's what the last point is. So effectively function in conjunction with the VRG to control breathing. So there's basically just intercommunication between VRG and DRG. So you go back to this diagram right here, okay? The DRG, the dorsal respiratory group, located at the dorsum posteriorly in the medulla, receives innervation from the vagus and glossopharyngeal nerves. Again, you know this, like vagus is cranial nerve number 10 and glossopharyngeal is cranial number 9, right? Okay, so you receive inputs from these two nerves, okay, over here. These nerves, where do they come from? We're going to talk about these later when we talk about mechanoreceptors or central and peripheral chemoreceptors, right? Okay, they come here, they integrate into the dorsal respiratory group, and this communicates with the VRG, okay? There's also this line going up here to the pneumotaxic center. We'll talk about this in a minute, okay? So ventral respiratory group, this is located ventrally or anterior in the medulla, again, with the dorsal respiratory group. That's why they're both the same color, okay? Efferent innervation to the respiratory muscles. We talked about what the efferent innervation is, okay? This also includes, by the way, phrenic nerve, right? So you talked about this in the respiratory system anatomy. It's the phrenic nerve innervates the, anyone answer? Anyone? Phrenic nerve? The diaphragm, right? Okay, diaphragm, all right. So efferent innervation from the phrenic nerve to the diaphragm, okay? And intercostal nerves, which basically go to near the ribs and stuff. So you talked about this in anatomy. The ventral respiratory group controls both inspiration and expiration and regulates basic rhythm, okay? Usually, when we're talking about accessory muscles, the ones that go against resistance or during exercise, we talk about VRG, okay? It's involved in cardiorespiratory coupling and bronchial muscle cells and also in high altitude and exercise, okay? So with high altitude, when you're like hyperventilating or hyperventilating, okay, this is included with the VRG. It's composed of three cell groups: the rostral nucleus retrofacialis, caudal nucleus retroambiguous, and nucleus parambiguous. Honestly, you did not go into details of this in your recording, so I don't think you need to know these, but it's good to know, okay? He actually skipped through most of these. He went mostly over the last section of this lecture. That's what's mostly important, okay? So how did the DRG and VRG work together? So what's happening here, this intercommunication, okay? Sorry, does anyone have a question?
Nope, apologies.
No worries, it's okay, all right. So the VRG, or the ventral respiratory group, contains inspiratory and expiratory neurons, okay? So these we just talked about, involved in basic rhythm generation, okay? So the normal breathing that you do, which the physiologists call quiet breathing, okay? That's also included in VRG, plus mostly the accessory muscles that we talked about earlier, okay? And the phrenic nerve and intercostal nerves are also innervated by these, okay? So the phrenic nerve going to the diaphragm, the intercostal nerves going to the ribs. That's how it's basically involved in quiet respiration, okay? They can send repetitive bursts of inspiratory signals independent of afferent innervation. So basically, if you sever these right here, okay, the vagus and glossopharyngeal nerves, basically cut them, okay? You won't have any afferent innervation, but still the VRG will still somehow maintain its level of breathing, okay? That's basically like, a protective mechanism. So impulses fired across the cranial nerve number 9, which is glossopharyngeal, and 10, which is vagus, will stimulate the DRG, okay? So that's basically the integration of inputs I just talked about. And then the DRG and VRG are involved in communication to generate a ramp signal. So you know how a ramp works, right? It starts here at the bottom, goes slowly up the top. Basically, the idea of it is that you get more and more impulses, okay? The frequency increases. That's how you continuously inhale, basically, okay? Until a certain point where you have to stop inhalation. And who does that? That's the pneumotaxic center, okay? There's a lot of P's here, okay? So the pneumotaxic center is located posteriorly in the upper pons, okay? in the nucleus parabrachialis. Okay, you don't need to know this part, but it's like posteriorly in the upper pons. So hold on one second. All right. Up here, okay? That's where the pneumotaxis center is located. So the DRG, while it's intercommunicating with VRG, it also has this one end going to the pneumotaxis center. When you want to basically switch off inspiration, okay, that's what's said here, and you want to control the rate of or frequency of breathing, basically, okay? So why is frequency of breathing important? That's what decides whether you're going to hyperventilate or hypoventilate, right? So hyperventilation, you breathe really fast, okay? Hypoventilate, you take deep breaths, basically, right? It switches off the ramp signal, which was generated by the DRG and the VRG. All good so far? Everything clear? I hope so. All right, let's move on. Okay, the next control of breathing is dependent on mechanoreceptors, which are involved in the pulmonary reflexes. Okay, so you have three right here: slow adapting stretch receptors, fast adapting stretch receptors, and C fibers or J receptors. Okay, so first of all, your basically how your body works is if you continuously breathe, if you continuously inspire, right, you're basically going to like burst, right? Your lungs continuously expand, eventually you're going to burst. Okay, so your body is like that. It's going to be like wait and got it, and it's going to have these certain stretch receptors which are going to detect you're inspiring too far, okay, and then it's going to basically stop inspiration, so terminate to inspiration to a certain extent. Okay, so these first set of receptors, which are called slow adapting stretch receptors, the way they work is, first of all, they're located in the airway smooth muscles. So if you have bronchial smooth muscles, okay, that's where they're located. The name tells you everything, right? So when they stretch, these receptors are activated, basically. Are activated, they send fire, they fire impulses, basically, and they slowly go back to normal, okay? Innervation of these receptors is myelinated vagal fibers. So we're talking about the vagus nerve. Again, this vagus nerve will integrate into the DRG, and that's what's going to decide, like, the rhythm of breathing. So wait, let's go back here. Okay, the vagus nerve, which goes to the DRG, okay, and then we talked about the afferent input from this mechanoreceptor, the slowly adapting stretch receptors, they go to the rhythm generator. There they act on the DRG. There's efferent output to the respiratory muscles, and that's basically the feedback loop they're talking about here, right? Okay. All right. So what are these receptors activated by? Again, the name tells you everything. So stretch, okay? So lung distension, when your lungs expand during inspiration, that activates these receptors. If you hold your breath too long, okay, or if you deflate below FRC, okay, functional residual capacity, that was covered by Zayad during his lecture on lung volumes, right? So that's basically expiratory volume and residual volume. If you go too far below this, okay, so basically your tidal volume is increased, right? That's going to also activate these receptors. So you don't like expire too hard or inspire too hard, okay? That's basically the mechanism of action of these receptors. So it's an inspiratory terminating, or basically it stops inspiration, or expiration to a certain extent, okay? And this is through negative feedback. So again, feedback's important, right? Next, this is basically an image showing you how this works. So your rhythm generator, it's like sending efferent output to tell you to inspire. Your inspiratory muscles, they're receiving positive input. They're going to inhale basically air, okay? That's going to cause lung distension. So these slowly adapting stretch receptors are going to send negative feedback. They're going to tell it to stop, right? Okay. Rapidly adapting stretch receptors. Basically, their location is in the airway epithelium. If you want to compare it, here it's airway smooth muscles, right? That's one of the differences. The innervation is the same. It's myelinated vagal fibers. Again, you remember from FND, myelination means faster impulses. So that makes sense too, right? They're activated by irritants or lung distension, okay? It's the lung distension part's the same as slow adapting, but like the irritants is more important here. So basically, they cause a cough reflex. So if you have allergies, if you have something stuck in anywhere in your respiratory system, okay, this airway epithelium will detect it. Okay, these rapidly adapting stress receptors will detect it. Myelinated vagal fibers will carry an impulse again to the DRG, the dorsal respiratory group. Okay, these irritants are basically going to activate these new receptors, right? Those nerve endings will go to the dorsal respiratory group that will mediate, breathing. So that rhythm will change. That's going to cause the cough reflex, gasping, and bronchoconstriction. Dr. Shams mentioned in your lecture that if someone has a lung transplant, since they don't have these myelinated vagal fibers innervating these receptors, you won't have the cough reflex. Okay, that's a bit important. So if someone has a lung transplant, their rapidly adapting stretch receptors are kind of useless because they don't have these myelinated vagal fibers going to the nerve, to the lungs, and from the lungs to the dorsal respiratory group. So that's important, okay? Remember that. C fibers are J receptors. In past years, this was a really, really important subtopic, but this time he kind of skimmed over it. So I don't think it's going to come, but still, pay attention, right? Their location is near capillaries, okay? Innervation, this time it's non-myelinated. So the first two, slow adapting and rapidly acting, were both myelinated vagal fibers. This one isn't, okay? They're activated by an increase in interstitial fluid in the lung. So basically, that's edema. I hope you took this in the CVP lecture in hemodynamics, right, with SAD. And also pulmonary embolism, okay? So like you don't need to know the details of how a pulmonary embolus works. You're going to take this in POD anyways later, okay? But basically, it's like a blood clot that moves is called an embolus. If this gets stuck in one of your pulmonary capillaries, okay, you basically that capillary is useless. You don't get the oxygenation, right? So right to left shunting and whatever, which you took in ventilation and perfusion, right? That was yesterday's lecture, I think. This can cause tachypnea. So tachy means fast, pnea means breathing. Okay, so quick breathing and hyperventilation, right? And the function is rapid, shallow breathing. Okay, bronchoconstriction, cardiovascular depression. Okay. Anyone has any questions? Because we're done with we're done with mechanoreceptors. This does not apply to the PAL session, by the way. No questions? Okay, moving on. All right, the last one, which is the most important one. During your lecture, Dr. Shams spent a lot of time explaining this, which is why I think this is what's most likely going to be tested during your exam. So please pay attention. Okay, big diagram. You don't need to know these names, Lashki, Shtaki, and Michelle, okay? You just need to know there are three regions, the rostral area, intermediate area, caudal area. We're talking about chemosensitive regions. So basically we talked about the mechanoreceptors, right? Those were involved in feedback, and so are these peripheral and central chemoreceptors. We're starting with central, okay? He likes to call them chemosensitive regions because they're located like superficially. We're talking about the medulla oblongata, by the way, okay? They're located superficially, but you don't see the receptors exactly. They're basically like regions. So that's why they call them chemosensitive regions, okay? The three regions, the rostral area. Rostrum is like an elevated surface. So that's how you remember this is like on top. Intermediate area in between, caudal. So you have a cranium and a caudum, right? You took this in embryology. So caudal is like below, okay, at the bottom. You don't, again, I don't think you need to know all of these, but it's fun. Yeah, good to know. All right. What are these? These are the locations of the central chemoreceptors, right? One second. Sorry, it's my fault. Central. Okay, there we go. Okay, your central chemoreceptors are located here, all right? So location is in the medulla oblongata, in the brainstem, in the central nervous system, medullary raphe, and ventrolateral regions. That's a lot of text, but basically, this is the medulla oblongata, okay? This is a medullary raphe, and these are ventrolateral medullary regions. This is basically where the central chemoreceptors are located. I don't think he'll ask you about this, okay? But, you can give this as a hint in a question. So I guess it's good to know. Three groups of neurons located in the following areas of the medulla. Again, I don't think you need to know this. Activated by increased hydrogen ions. This is important, okay? He specified this. He focused on this a lot, okay? So increased hydrogen ions basically means a low pH, all right? How do you have an increase in hydrogen ions? Okay, so basically carbon dioxide buildup. Carbon dioxide is an acidic gas. You should know this from chemistry, right? So if carbon dioxide builds up, you'll have an increase in hydrogen ions. You'll have a low pH, and these hydrogen ions are what's detected by the central chemoreceptors. Not really the carbon dioxide that's being detected here, it's the pH, okay? And you'll see this in a second, okay? All right, what else? It's inhibited by cold. Okay, so if you have a person who has hypercapnia is basically an increase in carbon dioxide. Again, this is what's detected by the central chemoreceptors. Okay, so if you like put them in a cold environment or something, they'll start breathing normally. Okay, or if you give them anesthesia, which is a sedative, okay, that basically calms down the entire body. That's going to also like calm down the central chemoreceptor. Okay, so with an increase in partial pressure of carbon dioxide, okay, you'll have an increase in hydrogen ions, which decreases the pH in CSF. The CSF is called the cerebrospinal fluid, okay? And that's why the tidal volume, which is VT, and FR is basically the frequency of your breathing, both are increasing. So basically, this means you're hyperventilating, right? Okay. The reason you're hyperventilating is, again, to decrease the carbon dioxide, increase oxygen, okay? They are not stimulated by hypoxia. He mentioned this like 10 times during your lecture. So if he does not ask you directly about this, and he brings an MCQ on this lecture, which is most probably going to be on chemoreceptors, this is going to be a wrong option. He's going to say central chemoreceptors are activated by carbon dioxide and oxygen, okay, which is wrong. They are not stimulated by hypoxia, okay? Oxygen has no effect on these chemical chemoreceptors, the central chemoreceptors, very important. There's no immediate effect by hydrogen ions. One of the reasons is there's a certain level of buffering by hydrogen carbonate. So I think you took buffering in blood gas transport, right? And then the second reason is hydrogen ions are polar, okay? They don't pass so easily into the CSF. Okay, that's where the detection is by the central chemoreceptors, right? Okay, the function of the central chemoreceptors is basically negative feedback to return the body to normal, okay? So if you have hypercapnia, again, increased carbon dioxide, you'll have a decrease in pH, okay? So you're going to start breathing faster, hyperventilating, so that you increase the levels of oxygen, decrease the levels of carbon dioxide, you return to normal, okay? When we're talking about normal resting ventilation, how do you like how do you keep the resting ventilation at like normal, right? That's basically these central chemoreceptors. They're going to detect the pH, and based on that, they're going to like stimulate or inhibit the respiratory centers. Again, we said these were involved in the feedback system, right? Okay, if you have a problem with the hydrogen ions, right? If you have a problem detecting these hydrogen ions, these are the following conditions that can occur. It's good to know, right? So sleep apnea, when you're sleeping, you can't breathe. So A means no, apnea again means breathing, okay? When you're sleeping, you can't breathe. That's sleep apnea. Panic disorder, okay? Epilepsy, basically seizures and stuff, okay? And migraines, which are headaches. SID is basically sudden infant death syndrome, okay? And yeah, those are like those are very important. But you know, the most important thing you need to know from this slide is that these chemoreceptors are not stimulated by hypoxia. Oxygen has no effect. They sense the changes in carbon dioxide through hydrogen ions. So these are a receptor for hydrogen ions, which is dependent on the carbon dioxide levels. All right. They can cause hyperventilation and stuff. The rest of this is good to know as a physician, but like from a testing perspective, what's going to come in your exam, it's most likely going to be from this part right here, okay? All right, we're done with central chemoreceptors, okay? These are peripheral chemoreceptors. So let's look at this, all right? So you studied this in cardiovascular parts of the block, right? You have the aorta, the arch of aorta, you have like carotid arteries, external and internal, all right? Over here, you have an aortic body, okay? You also have the carotid bodies, right? Near the carotid sinuses and whatever. These are basically, like Dr. Shams already emphasizes on them, these are basically like really small organs, okay? With the highest like blood perfusion per like gram of tissue. Okay, so like a lot of blood basically passes through them. Okay, compared to how small the tissue itself is, a lot of blood passes through them. That's basically what he meant. Okay, so aortic bodies and these carotid bodies, they basically contain the system of type 1 and type 2 cells, which is right here. Okay, this is basically looking into the carotid body, which is called glomus caroticum. You don't need to know this. Okay, you have a type 1 cell and a type 2 protective cell around it. Okay, the mechanism, I'm going to discuss it in a second. It's like, first off, look here, there's afferent innervation, again, glossopharyngeal nerve, vagus nerve, okay? And yeah, the rest of this was covered in anatomy, so I'm not going to go into details of this. Peripheral chemoreceptors sense changes in the carbon dioxide levels, pH, and arterial oxygen. So this is important. See, peripheral chemoreceptors do detect oxygen, central do not, okay? When are they activated? When carbon dioxide levels increase, okay? So just like your chemical chemoreceptors, central chemoreceptors, which detect carbon dioxide increase through the pH, okay? This one directly detects carbon dioxide increase, okay? The pH decrease as well, and also arterial oxygen decreases. So if you're in a state of hypoxia, which is low oxygen in your blood, okay, these receptors will be activated. So here are the values, okay? What Dr. Shams will do is, if he wants to test you, he's going to be like, okay, partial pressure of carbon dioxide is increased to, I don't know, 50, 60, okay? He just wants you to know that that's above normal. For oxygen, like significant differences are below 65, okay? And for pH, if it decreases below 7.4, which is about normal for blood, considering all the buffers and stuff, okay? That's also going to stimulate these receptors. So this is important, okay? So type 1 glomus cells primarily sense arterial oxygen. We're going to talk about this in a second. But like the function of peripheral chemoreceptors is in hypoxia, hypercapnia, or acidemia. These receptors compensate by stimulating the glossopharyngeal nerve, which is cranial nerve number 9, okay? And the impulses are transmitted to the dorsal respiratory group, and this can cause hyperventilation, vasoconstriction, or tachycardia. So again, cardiorespiratory coupling was by VRG, okay? Again, the most important things you need to know from the side are when are they activated. This responds to arterial oxygen, okay? If there's hypoxia, peripheral chemoreceptors, not central, okay? The levels, it's good to know, like it's best if you do know. And that it responds to all three. For central, it responded to pH, okay? So hydrogen ions because of the carbon dioxide. But when they ask you, like, what does it directly respond to? It's basically hydrogen ions. See in a second, okay? All right. We talked about this right here. Basically, it's this slide, okay? So here you see a type 1 cell, okay? All right. So we're talking about peripheral chemoreceptors again. So if you have a decrease in pH or an increase in carbon dioxide, basically carbon dioxide is an acidic gas. So if this increases, you will have a decrease in pH, okay? So if either of these happen, or there's hypoxia, so you have low oxygen, okay? What's going to happen is if your pH intracellularly will decrease because of these two, or your hypoxia will directly induce this one, okay? So potassium ion outflux will occur, okay? Basically, that's what's shown here, all right? Potassium ion outflux occurs. Over here, as a result, just to balance it out, calcium ion influx will occur, okay? When this occurs, calcium ions, I think you know this from like synaptic transmission. Sorry, does anyone have something to say? Okay. So if you have calcium ions entering, okay, they're going to stimulate these vesicles. Okay, there's a neurotransmitter being released here, dopaminergic basically. So dopamine is going to be released here, and these are going to bind to receptors over here. That's going to stimulate an action potential. Again, action potential through, depending on whether it's a carotid body, which is going to stimulate the glossopharyngeal nerve, or an aortic body, which is going to stimulate the vagus nerve. Okay, these will send impulses to the dorsal respiratory group. And that's what's going to maintain the rhythm of breathing. Okay, right, and that's why you get hyperventilation, or whatever. Okay, now for peripheral chemoreceptors, low oxygen level, again, hypoxia in the blood promotes potassium ion efflux from type 1 cells, compensatory depolarization. This is basically what we discussed over here, right? That's basically written in text. Calcium ions will move from will move the vesicles containing dopamine to the plasma membrane near the nerve ending, and exocytosis follows, where the neurotransmitters are going to be released. They're going to bind to receptors here. An action potential is elicited. It's going to pass through the neuron. Okay. So again, action potential is sent to the DRG to compensate for hypoxia, which is low oxygen, or acidosis, which is an increase in hydrogen ions, or a increase in pH, increase in carbon dioxide, decrease in pH, right? Okay. These graphs, we spent about 15 minutes on them. So these are also really important. Again, the chemoreceptor part and the graphs, okay? He's most likely going to bring the question from them. So what happens is, if you have hypoxia, again, peripheral chemoreceptors are the ones that are going to detect it, okay? So let's look at this. Okay, so partial pressure of oxygen over here is pretty high. So you're not going to have a change in ventilation. It's going to be about here, like normal, right? When you decrease oxygen, that's basically promoting like hypoxia, right? When you decrease oxygen, okay, you're going to increase the ventilation because of the hypoxic response, okay? That's what's shown here. So your alveolar ventilation will increase. Now, with an increase in alveolar ventilation, talked about this, I think, in yesterday's class. When you increase alveolar ventilation, you're going to decrease carbon dioxide because you're increasing oxygen that's entering, you're decreasing carbon dioxide because you're expiring it quickly, okay? That's what's shown here. So carbon dioxide falls. I'm talking about the red line, okay? Carbon dioxide falls. Now, because carbon dioxide falls, the central chemoreceptors are going to detect this. They're going to counter whatever the peripheral chemoreceptors are doing. So the central chemoreceptors will detect, okay, carbon dioxide levels are low, okay? You're going into respiratory alkalosis because your pH is going to go high because you're getting rid of the acidic gas, okay? So it's going to basically counter this. Because of the decrease in carbon dioxide, your ventilation decreases, okay? Because of the decrease in ventilation, you're hypoventilating. Your carbon dioxide increases. And again, you're going to start ventilating again, right? Because you're basically reaching hypercapnia again, okay? Over here in the green line, you're basically keeping the carbon dioxide levels constant, okay? If you have carbon dioxide levels constant, you're never going to reach this decrease right here. So with an increase in alveolar ventilation, you're going to increase with a decrease in like the oxygen, right? Partial pressure. With a decrease in oxygen partial pressure, you're going to increase the ventilation. That's basically what's shown here, okay? Carbon dioxide levels are constant. So there's no counter inhibition by the central chemoreceptor because they will never detect a change in carbon dioxide. The only one that detects the change in oxygen, again, is the peripheral one. Okay, three graphs here, okay? They're basically referring to the same thing. So we're looking at ventilation on all of these y-axes, okay? You have X on the x-axis, you have different variables. So over here, you have carbon dioxide, pH, and arterial oxygen, right? So chemical stimulus for breathing. If you increase carbon dioxide, you're going to cause hypercapnia. That's going to stimulate chemical chemoreceptors, which are going to go to DRG. Those are going to increase the ventilation. So you're going to get hyperventilation up until this point, where Dr. Shum says it desensitizes the neurons. So you're no longer going to have an increase in ventilation. It's going to decrease from this point forward. Okay. Over here, if you decrease pH, okay, again, decreasing pH is detected by central chemoreceptors, also peripheral, but like mostly central. They are going to respond to, oh, by the way, yeah, central chemoreceptors, they respond to 80% of this pH and carbon dioxide change, okay? And 20% is by peripheral, okay? That's also important. So that's going to come in the next slide, okay? So your pH decreases, your pH is falling. That means an increase in hydrogen ions. Again, this is detected by the central and peripheral chemoreceptors. They're going to cause hyperventilation. They're going to increase ventilation. But this response is similar to the hypoxic response, which we talked about right here. Because again, you're going to reach alkalosis, okay, when you try to increase the ventilation, and that's going to like cause a counter inhibition right here. If you keep it constant, if you keep the carbon dioxide constant, and you're going to increase ventilation, you're going to get, uh, you're going to get a higher level of, yeah, you're going to basically get rid of carbon dioxide. You're going to increase oxygen levels, okay? So it's going to be like a straight line, linear kind of here, okay? Here you have a bit of a curve, same thing that we just discussed over here, all right? So, yeah. Does anyone have any questions so far? Okay, yes, no. Okay. Again, this is important, okay? What we talked about earlier, 0% response by central chemoreceptors, okay? This is probably coming as a question, by the way. So 0% response by central chemoreceptors, 80% of the carbon dioxide response is by central chemoreceptors, okay? Again, carbon dioxide response is indirect, okay? So it's going to cause an increase in hydrogen ions. That's going to decrease the pH. That's going to cause hyperventilation. Over here, you have peripheral chemoreceptors. They're going to respond to 100% of the oxygen. So whenever you have hypoxia, it's basically these receptors, not these ones, peripheral, okay? 20% of the total response to carbon dioxide is by peripheral, okay? So it's less than central. And these respond to all of them, carbon dioxide, hydrogen ions, oxygen, okay? This one responds mainly to hydrogen ions, which was again to carbon dioxide, okay? All right. Over here, he's talking about acute versus chronic hypercapnia. Again, hypercapnia is an increase in carbon dioxide. Acute means for a short time, chronic means for a long time. So he increased carbon dioxide levels from 40 is normal, okay, 40 to 50, and then he kept it at 50 throughout this entire interval, okay? So acute increase in carbon dioxide would increase the ventilation. Why? Because both peripheral and central chemoreceptors are being stimulated. Because as I told you before, carbon dioxide influences hydrogen ions. That's going to cause the 80% increase in 80% of the total response to carbon dioxide and 20% by peripheral. So you're going to increase this basically right here, okay? All right, and then it goes down. So these peripheral chemoreceptors are only stimulated, okay? It goes down to this. So this, let's consider this part right here as 100%, right? This part right here is about 20% of this, which means that only peripheral chemoreceptors are stimulated, because we talked about this right here, like 20% of the response is by peripheral, okay? So that means central, for some reason, stopped. Why is that? So in the CSF and blood, your pH will basically, due to the chronic hypercapnia, your pH will be low, right? Your pH will decrease, your carbon dioxide will be increasing. Now, what the kidneys do, and this is basically relevant to renal, okay? And Dr. Shams also teaches renal, so he might bring a question on this part, okay? Your kidneys try to compensate for this decrease in pH. What they do is they try to either excrete hydrogen ions or they increase hydrogen carbonate, okay? That will basically bring pH back to normal. What did we say central chemoreceptors respond to? It's pH, the hydrogen ions, right? If the pH is brought back to normal, these have no reason to be stimulated. But your carbon dioxide levels are still high, okay? Your carbon dioxide levels are still high, and that's what's being detected by the PCR. That's why you're still getting a response here. That's why it's not back to normal ventilation. It's a bit higher because peripheral chemoreceptors are still responding to the high levels of carbon dioxide because we had two problems here, high levels of carbon dioxide and low pH. Okay, central chemoreceptors basically responded to the low pH, and with the kidneys fixing this problem, they stopped working. So only peripheral chemoreceptors are going to work now. And that's why you get this curve right here, okay? Acute versus chronic hypoxia. So as you can see here, if you have acute hypoxia, you're not going to have like a immediate steep curve that's going to go reach plateau really quickly. No, you need some time to get used to normal. The problem is with acute hypoxia, okay, you're going to stimulate again peripheral chemoreceptors, okay? You're going to increase the ventilation, right? You're going to reach alkalosis. So what's going to happen is uh, the alkalosis from the hyperventilation, because of the peripheral chemoreceptors, will inhibit response to hypoxia. Okay, so the central chemoreceptors will be like, okay, the pH is normal. Okay, so they'll tell them to stop now. So peripheral chemoreceptors will no longer work during this time. It'll take some time to get to this plateau right here. Okay, also this is important in like high altitude areas, okay, because you're going to hyperventilate and stuff, okay. In high altitude areas, you're also going to promote erythropoiesis, which is an increase in the number of red blood cells, right? And that's also going to help you adapt to this environment. So what he likes to give as an example is there are climbers that want to go to the Mount Everest. They'll camp at certain points until they reach the top. They won't directly go to the top because they don't acclimatize really quickly. They don't adapt to the environment. They basically, fall or something. Okay, big diagram. But we talked about the musculoskeletal. Oh, we didn't. Okay, we'll go back to the first slide, right? But musculoskeletal system is going to stimulate the sensory receptors. Let's go over this one by one. Okay, mechanoreceptors, the musculoskeletal system, when you are working, okay, it's going to stimulate the sensory part of the cerebral cortex. Okay, the motor part of the cerebral cortex will send messages to the rhythm generator, which is again the respiratory center. That's going to go to the respiratory muscles, right? This motor part also goes to the working muscles, right? And because these are under your control, these are the musculoskeletal system. This is like voluntary controlled skeletal muscles, right? Here's stimulation to feedback. So mechanoreceptors, which are basically responsible for, whenever you're moving, whenever you're working. We're also talked about mechanoreceptors with lung distension and, slowly adapting receptors. So those are also going to prompt the rhythm generator act on the respiratory muscles. That's basically stimulation through feedback, okay? The direct stimulation, okay, so wait, this is easier to explain on the first slide. Here we go. Okay, so this is what we're talking about. Mechanoreceptors in the musculoskeletal system. These will give a without feedback signal to the rhythm generator. That's going to affect the respiratory muscles. There's also these mechanoreceptors which send a message to the rhythm generator. Those are going to act on respiratory muscles, but these are with feedback. Okay, and of course, cortex exercise and voluntary, movements. These are going to act on the rhythm generator again. Okay. All right. Okay. Yeah, this is the last slide. Okay, so maximum voluntary ventilation is a clinical pulmonary function test used to assess your overall respiratory apparatus function, which is the highest one here. So your highest ventilation is because of this pulmonary function test of maximum voluntary ventilation, because you're controlling it. This is through respiratory apparatus. I think you talked about spirometry and stuff with Zayad again. So that's what gives you the maximum ventilation. The second one would be exercise, okay? And then you have the PCO2, pH, and PO2. All right. Again, PCO2 is high because both peripheral and central act on, right? Does anyone have any questions? Is anyone still here? Okay. Right. I have some questions. If someone's still here, they can answer, right? They can type in the chat box where I'll get the responses, right? First question. A 16-year-old with head injury from a car accident was brought to the emergency department by an ambulance. On exam, she had a respiratory rate of 8 breaths per minute and a prolonged duration of inhalation was noted. Damage to which of the following groups of neurons in the brainstem is most likely responsible for this abnormal respiration? Anyone can raise their hand and then unmute and answer.
Is it A?
A? Okay, wait. So what they're going for here is the person has damage to a set of neurons which is responsible for this abnormal respiration, which is basically prolonged duration of inhalation. He does not stop inhalating. She does not stop inhalating. What's the respiratory center group of neurons that's responsible for stopping inhalation, basically? That's what they're asking. Okay.
It's C, right?
Yeah, it's C. Good. Okay. So the answer is C. Okay. And the reason for that is they're talking about the pneumotaxic center. So basically, we said switching off inspiration depends on the pneumotaxic center. Okay. Why are the other options wrong? Dorsal respiratory group. This receives innervation from the mechanoreceptors, central and peripheral chemoreceptors, right? And that's what's going to govern whether you're going to breathe fast or breathe slower. But it's not going to determine the inhalation process. It's going to just integrate the inputs, right? So there's that. Ventral respiratory group is for normal basic rhythm, okay? The ramp signal and all that, but it's not directly responsible for stopping inhalation. Vasomotor center, again, that's higher center, brain cortex and stuff. You don't need to know this, but like, not for this lecture specifically, but I think it's mentioned in your integration lecture. And the nucleus tractus solitari, I told you dorsal respiratory group is in this set, right? Nucleus tractus, so that's why it's wrong. All right, next question. Concerning the respiratory centers, which of the following is correct? For this question, I suggest using elimination, by the way. It's better.
Is it D?
Good, it is the, okay. So, nice. Okay, so why are do you want to go over why the others are wrong, or it's your choice? I can go over them too, but like, if you want to go over why the others are wrong, it's fine.
It would be better if you explain.
Yeah, okay, sure. Okay. So the normal rhythmic pattern of breathing originates from neurons in the motor area of the cortex. No, cortex was the higher center, okay? It's not the higher centers, it's the respiratory centers, okay? That determines the rhythmic breathing. During quiet breathing, expiratory neurons fire actively. This is wrong because I said inhalation is an active process. Expiration is passive unless you're like breathing against resistance or exercising. So quiet breathing, which is normal breathing, you don't need expiratory neurons firing, which were in the ventral respiratory group, by the way. Impulses from the pneumotaxic center can stimulate inspiratory activity. We said this is wrong. We covered this in the last question, which is they basically switch off inspiration, right? This is the correct answer. Only output from the respiratory system is via the phrenic nerves. No. Although this is one of the outputs, it's not the only output, right? Phrenic nerve is for the diaphragm. There's also intercostal nerves and stuff. So, yeah. Next question. Okay, this one's good. Which of the following receptors activated by irritants, the lungs, and what is the effect of the receptor activation?
C.
C. Yeah, C is correct. Nice. All right, so why are the others wrong? Do you want to go over them, or should I? It's fine if I can go over them. It's great.
Yeah, you can.
All right, sure. Slow adapting stretch receptors don't induce cough reflex, right? Slow adapting stretch receptors do not induce rapid shallow breathing either. This was the function of the C fibers or J receptor, okay? These are involved in basically terminating max inspiration or max expiration, okay? Which is this right here. So rapidly adapting stretch receptors induce cough reflex, yes, okay. This is wrong again, and J receptors again wrong. They can add 6 more columns to this by adding they can add 6 more like rows to this by adding another column. They'll add myelinated, unmyelinated, okay? So rapidly adapting stretch receptor, like I said, is myelinated, and so is slowly adapting stretch receptors, but J receptors aren't, right? Okay. Anyone wants to give this a try? Concerning the central chemoreceptor.
C.
Very good, C, yeah, exactly. Okay. And this is a type of question that I think could come if he brings it in MCQ, he'll probably change some of these options around. But this is very important. Okay, so they are activated by changes in pH of the surrounding extracellular fluid. Which is correct, yes. Why does the change in pH occur? Because of the carbon dioxide. Ibrahim, you want to explain or should I go in?
It's fine. You can go over.
Right, sure. So they are located near the dorsal surface of the medulla. Again, no, they respond to both the PCO2 and the PO2 blood. Like I said, this is the type of option you would bring. This is wrong, right? Which one does both of these?
Peripheral.
Peripheral, good. Yeah, peripheral. It responds to this, and hydrogen ions, right? For a given rise in PCO2, the pH of cerebrospinal fluid falls less than that of blood. Actually, it's more. Okay. This is more relevant to renal. It has to do with buffers and stuff, and that's also the fourth option. Fifth option, the bicarbonate concentration of the cerebrospinal fluid, that's CSF, cannot affect their output. It can. Okay, because the bicarbonates will buffer the hydrogen ions. You took this in blood gas transport, I think. So the bicarbonate will buffer the hydrogen ions, okay? And that's going to stop the change in pH, basically. And that's going to inhibit the central chemoreceptors. So it can affect their output because it affects their activation. Okay, I think this is yeah, that was the last question. Okay, this is a summary slide I found on Google. I think it covers most of it really well. So higher brain centers, such as the cerebral cortex, voluntary control over breathing. Other receptors, such as they will detect pain and stuff. They'll emotional stimuli act into the hypothalamus. I mean, these are for the higher centers, right? But for the respiratory center specifically, you have slowly adapting stretch receptors in lungs, irritant receptors, which is again rapidly adapting stretch receptors. Both of these are myelinated vehicle. Okay, this one was in airway smooth muscle. This one was in airway epithelium. Peripheral chemoreceptors, they detect oxygen, carbon dioxide, and hydrogen ions. This one detects carbon dioxide. Carbon dioxide over here is fine, but it's mostly hydrogen ions, right? Not oxygen, again, okay? Receptors in muscles and joints, okay, again, they'll activate, that's the musculoskeletal, okay? I made a Anki deck for this lecture. There's like 35 slides in this Google slide presentation. There's 33 cards in the deck. So one slide, one card, basically, okay? Covers a lot of information, but should be doable, inshallah, okay? So it's also going to be on the academic aid drive, inshallah, okay? We are done. Thank you all for listening. Okay, if you can please scan this QR code, give your feedback. If you want more Ryan Gosling memes for the next one, inshallah, let me know. But yeah, we're done. Thank you.
Thank you so much.
Yeah,
Thank
of course, anytime. Thank you guys for attending. By the way, I know there's like 1 week left until your CVP final. Sorry for the reminder, by the way, but I appreciate you guys attending. Okay, I know 2 times speed would have been easier, but yeah. Thank you all for attending. Have a great day and good luck for your exam, by the way. See ya.